Okay, guys, today we have our last lesson of Chapter 2, Basic Chemistry, dealing with biochemistry. Now, this will only be a small portion of biochemistry as a whole, and we're really just going to focus on the major classes of macromolecules or biomolecules, as you may see them written sometimes. So let's start with carbohydrates. Now, carbohydrates contain carbon, hydrogen, and oxygen. It's our starches and sugars that we commonly call them, and they are classified according to size with our simple prefixes mono, di, and poly. So mono meaning one, di meaning two, poly meaning three or more. Monosaccharides are simple sugars. Disaccharides are two simple sugars joined together by what's called dehydration synthesis. We learned about that in the previous lesson. And then polysaccharides, which are going to be long branching chains of linked simple sugars. So we can see in figure 213, we have this monosaccharide right here on the left in green. And then in 213b, we have our disaccharides. So we have two of them joined together right here. Dehydration synthesis has occurred. And that is why we have our oxygen right here that's bonding the two together. Okay, so then in 213c, we see this long branching molecule. This is actually starch. Okay, so I think I've talked about celery being an example of a carbohydrate. The starch that's in celery, those long strings that are in there, that's actually a bunch of these molecules that are all linked together. Okay, so dehydration synthesis is reversible. We already know that. Okay, so we can hydrolyze, so to say, a carbohydrate. We will separate it into those individual sugars. So we can see that here with dehydration synthesis, we pull out a water molecule and we take glucose and fructose and we form sucrose. That's table sugar. And then we can reverse it. We add water here and we split it. Hydrolysis, hydrolysis, water splitting, and we can separate them back to glucose and fructose. All right, so let's talk about lipids. Now, lipids contain carbon, hydrogen, and oxygen. They contain a lot more carbon and hydrogen than oxygen. They are insoluble in water. Now, we should all be familiar with what happens when you mix oil and water. They simply don't mix. You could shake it up all day long, stir it, and they're never going to truly dissolve like you would see sugar or a carbohydrate in water. So common lipids in the human body are neutral fats. These are triglycerides. We'll see based upon the structure why they're called triglycerides in just a minute. They are found in fat deposits within the body. They are composed of fatty acid chains and a glycerol head. And they are also a source of stored energy. Okay, so we can see right here this formation of a triglyceride. So we have these fatty acid chains right here, 1, 2, 3. And we have these OH groups, these hydroxide groups hanging out here. We pull those water molecules out. And once we do that, we form this neutral fat right here. This is how we store this energy within the body. So when you hear people say, if you want to lose weight, you need to drink a lot of water and exercise. Well, you need those water molecules to reverse this, break it down, and get the fat out of the body. Okay, so other lipids, the phospholipids that we find in the cell membrane. I'm sure that you guys remember that from Biology 1. And also steroids. Now, steroids include cholesterol, bile salts, vitamin D, and some hormones. They serve an incredibly role in communicating within the body. Okay, so this phospholipid right here, what we have done is we have partially unsaturated our one of our fatty acid chains, kicked another one off, and added this phosphorus-containing group right here. You can see we have a phosphorus here, which gives it a polar head, so to say. So that makes it water-loving. Okay, so cholesterol that we see right here, the basis of all steroids within the body. Now, all of our cholesterol chains are the same, except for this one R group right here. And also, I want to point out they are planar, meaning they are flat. So if you take this large structure right here and you turn it on its side, it's only going to be one atom thick. Okay, so this is why it's important to regulate cholesterol within your body, because this chain out here, or the R group that may be attached, can very easily get caught on the linings of your arteries. Okay, so let's talk about proteins real quick. Now, proteins are made of amino acids. There are 16 different amino acids. They all contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur in the R group. So the generalized structure of all amino acids is going to be an amine group. You may see it as an amino group. The acid group over here, also called the carboxyl group. Sometimes it's good because it's carboxylic acid. And then an R group right here. So some are very simple, like glycine. This is the simplest amino acid. It's R group, the functional group right here. It's just a hydrogen. Some of them are very complex, and we can see in lysine, some of them even more complex than that. Now, proteins account for over half of the body's organic matter. They allow us to make things within our body. They play a vital role in cell function, and they really have a lot of mass. Your muscles within your body, for example, they are pretty much just protein. That's it. Okay, your hair is made up of proteins. Now, they act as enzymes, hormones, and antibodies. And we've already talked about the amino acid structure that we see there, the COOH. Okay, that's carboxylic acid. That can also be represented as CO2H as well. And we have two types of proteins. Now, fibrous proteins, these are our structural proteins. They're found in our body structure. They include things like collagen, keratin. They are very, very stable. Okay. Now, one of the things that can change them is heat. So, if you get your hair straightened, that heat that's applied will cause the organization of it to change slightly, and more importantly, it'll cause it to keep that structure. Okay, now globular proteins. So these are functional proteins. They function as antibodies and enzymes. They can be denatured, meaning you can change them permanently. Now, this explains why when you cook beef, which is red when it's raw, it turns brown. Now, hopefully you're not cooking it too much and turning it black. Okay, so let's move on to enzymes. Now, enzymes are classified as biological catalysts. Now, they technically are proteins. Okay, they can increase rate of chemical reactions. Some of them very, very slowly. Some of them very, very, very quickly. Now, what they will do is they will come in. This substrate that's right here will fit with an active site. Now, once that occurs on a globular protein, it will induce a change. That change will allow the rate of a reaction to be sped up or slowed down. Okay, so they come in, they bind, and then they cause this change. Now, let's talk about nucleic acids. Now, I'm pretty sure you've all heard of DNA. That's what we see right here, deoxyribonucleic acid. So they provide this blueprint of life, so to say. They encode for our genes, which encode for whether or not we have, brown hair, blonde hair, blue eyes, green eyes, or even if we have dry earwax or oily earwax. Some of the most bizarre things you would never think of are actually determined by our genes. Now, every nucleic acid is going to be a polymer of what we see right here, this nucleotide. Now, a nucleotide is going to contain a phosphate group, a 5-carbon sugar, and a nitrogenous or nitrogen-containing base, which shown right here is adenine. But the 5 that we have that we're going to focus on in this course will be adenine, guanine, cytosine, thymine, and uracil. Okay, so let's talk about DNA, this double helix or twisted ladder model that we see right here. So it is organized by complementary bases to form a helix. Now, complementary means they pair together. So we can see thymine and adenine. They have this pointed shape right here as a demonstration, and cytosine and guanine have this curved shape. Now, thymine and adenine always pair together, and cytosine and guanine always pair together. Now, the other thing that you can look at very closely if you look at figure 219C is these little dots right here. Now, these dots actually represent the number of hydrogen bonds that are formed between the two. Now, adenine and thymine will form 2. Guanine and cytosine will form 3. Now, these hydrogen bonds that hold it together are pretty fragile. They're pretty weak, and they are easily able to be broken. Now, that's important because we have to copy our DNA between every cell division. Okay, so we need a complete copy of DNA in each cell before we start cell division. Now, another important one that we have to talk about is ATP, adenosine triphosphate. Now, this is the chemical energy used by all cells, and it's simply released by breaking high energy phosphate bonds, and then it is replenished by the oxidation of food fuels. So when we bring that food into our body and we break it down, that energy is then used to store in ATP. So again, we have ribose right here. Instead of deoxyribose, we have adenine up here. Okay, and then we have 1, 2, 3 phosphate groups right here. So in our nucleotide, that is just one phosphate group. So what happens is this: we break this bond right here, we release this phosphate group, and we produce ADP, adenosine di. If you remember from our discussion about carbohydrates, disaccharides mean 2, just like it is right here. Diphosphate means 1, 2 phosphates that are present. Okay, so figure 2.21 outlines what happens with all of this when we are going to use ATP to cause some type of change within our cell, causing, and figure 2.21 goes through step by step. We can see right here ATP comes in, and it will then open up this channel right here, allowing for, in this case, the solutes to be moved out. Okay, the same for our muscle cells. We use a lot of energy for them as well, and also for chemical reactions. Okay. So if you have any questions, make sure to post them in the comments below. You can send an email to mr.tucker.schs, or you can join the Google Hangout and post your questions there.